Hello everyone and welcome to the CellDesigner video tutorial. Today I will guide you through the construction of a 3-node network with a piece of software named CellDesigner that we have right here. If we just go ahead and click on the CellDesigner logo, CellDesigner will open on our screens. Now, what is CellDesigner? CellDesigner is a piece of software that provides a user-friendly interface for graphical biochemical pathway description. Of course, understanding the logics and dynamics of gene regulatory and biochemical networks is a major challenge for systems biology nowadays. But to facilitate this research topic, CellDesigner has been created to visualize these modeling networks that we create in the Modeling Immunity to Enteric Pathogen Center, or MIP. Now, CellDesigner will allow you guys to easily create networks using solidly defined and comprehensive graphical notation. Today, with this video tutorial, we will create a 3-node network describing Th17 differentiation. So let's go ahead and look at the parts of CellDesigner. In the main screen you will see the upper menu and you will have different options depending on the tab you're in. You also have a toolbar here, and the toolbar is composed of different parts. You have your node toolbar, and this will allow you to create nodes into the network. You have your reaction toolbar, and this will allow you to link, also to modify using different options. Then you have your compartments options that we will look later. This is your drawing area. So pretty much this is where you are going to see the action of your model, of your network. You have your nodes section in the bottom right side of the screen. You also can edit nodes and add nodes, and we will also get into that later. In this other part, we also have our list area, and in the list area we can see the different nodes we have, either species, proteins, genes, RNA, if it's sense or antisense, and also our reactions. We also have our layer area, but we are not going to get into that a lot. It's not a very useful part. What is useful is the tree area that I'm highlighting here. So as you can see, CellDesigner is a very useful piece of software, and it really helps us to construct and maintain the architecture of our modeling networks. So let's get started. Let's create a new network. Let's click File, New, and a new dialog will open. You're able to describe the name and the dimension of the network. On the name, let's go ahead and put T helper 17, or actually let's put Th17 3 nodes, and then we're gonna keep the dimensions like that. A recommendation though for big networks, keep those numbers in the thousands, because then it's really hard to go back and make a model bigger. As you can see, a new white square has appeared in our drawing area. This is where we are going to draw our network. The way we do it is by clicking these 2 compartment buttons here. As you can see, there are 2. We can create an oval shape. We can give a name. Let's put just example oval, and I'm just showing you guys the different shapes we can create. We can create the squared shape. We can also give a different name, but this will depend on really your model and the model you want to create. Let's delete those. Actually, we are going to construct our Th17 model by using a square shape. And what we will do is we will call it CD4 positive T cell, and we can call it -Th17, for example. This will be our cell membrane in our Th17 cell. Now, how do we start putting nodes in this network? If we check the species toolbar here, we can see there are different options. We can create a generic protein, for example. We can create a receptor, and as you can see, I'm giving names to each of those. We can create an ion channel that is in the cell membrane. For example, a truncated protein. We can also create that. Let's put here. Let's click on D. OK. For example, a gene, if we are modeling genetic networks. We can use the gene shape, RNA, or antisense RNA. It has a shape that is very particular to RNA. We can also create phenotypes, ions. For example, if an ion is coming out of a channel, we can model that. We can also create simple molecules if we are not really sure about what is it, or if it's just a compound that we are giving. We can create a drug. If we are giving a certain drug that upregulates a certain pathway, we can also do that. And as you can see, the drug has a double outer circle. We can create complexes, and that's extremely useful when you want to create a complex in the cell membrane. And you can drag and drop different species. And now, for instance, species G and E would be part of the complex I. Alright, so now that we've been over all these different types, and please feel free to play with those and pause the video and play with different types and different options. Now we're going to get started and create our Th17 differentiation model. Now, Th17 differentiation is mainly characterized by an upregulation of IL-17A, the cytokine IL-17A, and this is usually driven by a transcription factor named ROR gamma T. So here, if we click to the generic protein, we can start creating these reactions. The first protein that we want to create is the induction, which is TGF-beta plus IL-6. See how it's in here? Now the next protein we want to create is the transcription factor that it upregulates, which is ROR gamma T. We'll press OK. The next one we will create is the cytokine that ROR gamma T upregulates, which is IL-17. So following the same rationale, we're going to create a species named IL-17A. Now here we have our 3-node network, but these species aren't linked. There's no link from TGF-beta IL-6 to ROR, no link from ROR to IL-17. So how do we create those links? Here in the reaction toolbar, we have different options. We can create a state transition, for example, and please notice that you can select where do you want this reaction coming from. So if we click and then we select the final point, we can create this reaction. Now TGF-beta and IL-6 is linked to ROR gamma T. Now please note that there are other reactions. For example, we can create a transport, or we can create maybe a transcription reaction. So now you can notice, well, let me drag this here exactly. Do you see now the difference between different reactions, the different shapes of these arrows? This is different annotation, and again, feel free to play with this, pause the video, play with the reactions. But for our model, we are just going to use state transitions. So now let's create one from ROR gamma T to IL-17A. So here what we can see is that these reactions have been added, and the first species that we create becomes the reactant. The second species that we link the reaction to becomes the product. Now, let's say that we want to change some individual options. For example, the color or shape of this node. By right-clicking and selecting color and shape, we have a different array of possibilities. We can change the size, we can make it wider, for example. The line of the node, we can make it wider as well, and then we can choose a different color. For example, this yellow here. Let's press OK, and let's center this one. We got it wider, let's center it here. Perfect. Now we have our ROR. Let's change the color on the ROR, and let's select, let's see, green. Yeah, kind of this green. Let's make the line wider, just to be consistent. Oops, okay. Yeah, let's put just 2 and press OK. And let's also change the color of the third one, which is IL-17A. Change color and shape. Let's do a kind of like a reddish type. Yeah, that one would work. We'll also change this to 2 and press OK. See how this model now looks much prettier than before. Especially you want to be consistent with colors and shapes. You also can use the swatches. You have a lot of possibilities here, but you can also look for the color you like, or if you have the codes of RGB, you can also use that. We'll press cancel because we don't want to change the color. So here is the model that we have. We created a model of 3 nodes. TGF-beta and IL-6 upregulate the transcription factor ROR gamma T, and this transcription factor upregulates IL-17A. Now we can also create these active states. So what this means is that ROR gamma T is now active because there's presence of TGF-beta and IL-6. We can also click and just press the letter A in our keyboard, and do you see how the active state is created as well? This is how you create the active states, but this is pretty much how you create your TH17 network. Now if we press one node and we go to the bottom right side, we can actually add nodes. There are different nodes that are predetermined, but if we click edit nodes, we can write whatever message we want to write here. So let's say in the presence of interleukin 6 and transforming growing factor, let's put beta ROR gamma T is upregulated. Perfect. We can also add the reference where I took this information from. More specifically, I took this information from a Cell paper. I think the PMID is 16990136, and we can add that this is totally optional, but I like to add my sources of information. And these nodes, by pressing OK, now we're going to save it, but these nodes are carried in our computational model later. So it's really useful to have this annotation. If we ever want to make a change, for example, I want to put IL-6 instead of interleukin 6, and I want to make this TGF-beta, I want to abbreviate this TGF-beta and not having to write transforming growing factor beta. So we can add that, modify it, and press OK. So here you have your nodes. It's really useful for annotation, model annotation especially. This is a very useful tool. Now in our list part of the software, we have our species. Do you see here how we have protein 1, which is our TGF-beta and IL-6, protein 2, which is our ROR gamma T, and protein 3, which is our IL-17A. This is a list of the species we have. But more specifically, these 3 species are proteins. We have these 1, 2, 3 proteins, and you can see here the names. Genes, RNA, antisense RNA, we don't have anything, but this is because we haven't selected anything from the upper menu in the species toolbar. If we press reactions, we have 2 reactions, reaction 1 and reaction 4 here. Now please notice that this is not reaction 2, it's reaction 4, but if you remember, we created 2 other reactions to show you 2 other options that we have to link species. CellDesigner will always remember what we've done, so CellDesigner annotates accordingly. Only one compartment, the CD4 positive T cell TH17, the one compartment we created at the beginning, and that would be it. If we check our tree space, we see how we have 3 species and 2 reactions. Really, this is how you create your model. There's no more secret into that. If we want to save the model, we'll go to File, Save As. We can select the location. Let's say I want to save this here on the desktop. We can select a name. For example, that one, click Save. And also for publication purposes, we may want to get an image of our model. So File, Export Image is the option we need to take. Same thing, let's select the location and press Save. We have different options actually for images. We can do a PDF, GP, EG, PNGs. I usually use JPEG, so you can use that if you want. And here you can see in my desktop, now I have my image here. Perfect. And I also have my XML file here. Now, one thing I wanna make this point. When we go to File and we click Save As, I don't know if you've noticed, but seeing the format, how it says XML, SBML, XML, actually there, if we click there, there's no other formats. CellDesigner will save as an SBML, Systems Biology Markup Language, which is really useful also if we want to import this network into a modeling software like we will with Copasi.